This video goes along with your chapter 14 notes, which is our first big look at heredity. We will look at Mendelian genetics and non-Mendelian genetics, as well as environmental effects on phenotype and some on chromosomal inheritance, but we'll see more of that after or in the next chapter. We're going to look at Mendel's experiments first. So Gregor Mendel grew up on a farm, and then he entered a monastery, and he really liked plants. And he bred garden peas because he was curious about inheritance. And it turns out he was right about a lot of things that he figured out based on the experiments that he did. He used peas because there are many varieties. He called each feature a character and each variant of a character a trait. They're also good because they have a relatively short life cycle. They produce lots of offspring, and it's easy to control the mating between plants. He tracked characters that occurred in two distinct forms, for example, purple flowers and white flowers for flower color on a pea plant. He started the experiments with true breeding plants, meaning only the same trait is produced from parents every time. So if it has purple flowers, all offspring every time will also have purple flowers. When we talk about the mating of two true breeding varieties, we call that hybridization. True breeding parents are called the P generation. The offspring of the P generation are the F1 generation. The mating of two F1 plants gives F2 generation offspring, and he followed those traits for at least the P, F1, and F2 generations and sometimes longer. And it is a really good thing that he did, because if he had not gone that far, he would have missed a couple of very important things. All of his observations and research led to two fundamental principles of heredity: the law of segregation and the law of independent assortment. First, we'll look at the law of segregation. When he crossed two true breeding plants, one with purple flowers and one with white flowers, all of the F1 offspring had purple flowers. But then when he bred the F1 offspring by either self or cross-pollinating, in the F2 offspring, some of them had purple flowers, or most of them did, but there were some where the white flower trait came back. And he concluded that the trait or the white flower factor was hidden in the F1 plants. And that's kind of where he got his ideas for the dominant and recessive trait. So you can see that in the diagram or figure 14.3 on the right. He saw a similar pattern in 6 other characters that he looked at, including seed color, seed shape, pod color, pod shape, flower position, and stem length, which you can see outlined here. And if you'll notice, with those F2 offspring, the ratio of dominant trait to recessive trait is all about 3-to-1, which is supported by the statistics that we look at when we actually go through and do Punnett squares with this in a minute. He developed a model to explain the 3-to-1 pattern he saw in the F2 plants that is part of 4 key things, or has 4 key parts. The different versions of a gene account for variation in inherited characters, where we now call an allele a version of a gene. An organism inherits 2 copies or 2 alleles of a gene. If the alleles are different, the dominant allele shows. And also, finally, all of that put together, we get the law of segregation that says that the 2 alleles for a character separate during gamete formation, which we now know is because during meiosis, our homologous pairs separate, and then our chromatids separate in meiosis too. And we can use a Punnett square to predict offspring outcomes, and we will. Here is a picture version of the law of segregation. Before we move on, we're going to take a pause for some useful genetic vocabulary that I hope you will remember from Bio 1. A homozygote is an individual that has 2 of the same alleles for a character, and we would call them homozygous. A heterozygote has 2 different alleles for a character. A phenotype is the organism's appearance, so like what we can actually see. and the genotype is its genetic makeup. Moving on to the law of independent assortment. He derived the law of segregation from looking at one character, which we call a monohybrid cross. But then it took him following two traits at the same time to get to the law of independent assortment. And he knew which alleles would be dominant from all of the monohybrid crosses that he did. The law of independent assortment says that genes assort independently during gamete formation. So here is an example of monohybrid versus dihybrid crosses. So monohybrid, we're just looking at flower color. With a dihybrid, in this example, we are looking at seed color as well as seed shape. So yellow versus green and round versus wrinkled. And you can see that if they assorted dependently of one another, we would see a 3-to-1 ratio. But what he actually saw was a 9-3-3-1 ratio, which is where we get independent assortment from. Before we get into some Punnett square practice problems, we need to look at some probability laws that govern Mendelian inheritance. So we're looking at this, the same rules of tossing a coin or rolling a dice and drawing cards. Our probability scale ranges from 0 to 1. And there are both multiplication and addition rules that we'll need to know. And we'll start with multiplication. In statistics, we multiply things together when we want to know the probability of event 1 and event 2 happening. So an example, you flip two coins. What is the probability that they are both heads? So the probability that the first one is heads is 1/2. Probability that the second one is heads is 1/2. And we multiply that together to get 1/4. And we can extend that beyond 2 events if we like. So maybe we want to look at 5 traits, for example. So we would show the probability of each of those 5 things occurring to get to our final answer. In addition, we add when we have individual probabilities of one or two or more mutually exclusive events will happen. So maybe we want to know the probability that an F2 plant is heterozygous, the probability that they get a dominant allele from the egg and the recessive one from the sperm is 1/4. The opposite is also true. Put that together, we get 1/2. We can use the rules of probability to solve complex genetic problems. So we are going to use this to predict outcomes involving multiple characters. Starting with example 1. Let's say we have 2 plants that are heterozygous for both seed shape and seed color. Remember that yellow is dominant to green and round is dominant to wrinkled. And we want to know the probability that the offspring are also heterozygous. So there are several ways to do this, but I'm going to go with the easy way and multiply, because we will need to know how to do that when we get to looking at more than 2 traits. So what I'm going to do is set up 2 Punnett squares, one for seed color and one for seed shape. So both parent plants in this case are heterozygous for both traits. We'll just fill this in real quick. Okay. And we want to know that the probability that the offspring are also heterozygous. So let's look, I'm going to change colors here. Got a heterozygote there, and there. So I'm going to multiply the probability that they're heterozygous for both. So for seed color, that would be there's a 1/2 chance of being heterozygous for that trait. And it's also 1 out of 2 they'll be heterozygous for seed shape. 1/2 times 1/2 is 1/4. So there is a 1/4 probability that the offspring will be heterozygous as well. Moving on to another one. Same thing, we're starting with both parents are heterozygous, and we want to know the probability that the offspring are heterozygous dominant. So let's set up our two squares again. So let's find our genotypes that are heterozygous dominant, 1 and 1. So we have 1/4 times 1/4, which is 1 out of 16. And we'll skip this one because I think you get the idea, but please try this one on your own. The truth of the matter is that inheritance patterns are often more complex than what Mendel predicted. His description of inheritance patterns do not fit all organisms or all characters, and geneticists have spent the last several years expanding on these ideas. Well, and what we know is that not all traits are determined by one gene. The relationship between genotype and phenotype is not always so straightforward, and sometimes there isn't a dominant and recessive trait. So what we're focusing on from now on is what we call non-Mendelian genetics because it doesn't fit Mendel's ideas. So we are going to look at alleles that are not completely dominant and recessive, what happens when a gene has more than two alleles, and when a single gene produces multiple phenotypes. Starting with degrees of dominance. Mendel's ideas show complete dominance, where we have a dominant trait and a recessive trait, and that's it. If the dominant trait is present, it shows. If two recessive alleles are there, then that's the trait that shows. And for some genes, neither allele is completely dominant. And there are two things that could happen there. One is incomplete dominance, where the phenotype of the heterozygote is an intermediate between the phenotypes of the homozygotes. An example is that snapdragons can have red, white, and pink flowers, where pink would be the heterozygote. The other is codominance, where the phenotypes of both alleles are seen in the heterozygote. And we see that with human blood type, which we'll actually look at in a second. Another thing I want to make clear about the frequency of dominant alleles is that just because it is dominant does not mean that it is what we see most often in a population. All that means is that the character always shows when at least one dominant allele is present. An example here would be polydactyly, where an individual has extra fingers or toes. That is a dominant character or trait, but it only occurs 1 in every 400 births, which is higher than I thought it would be. But still, that's relatively low. Relatively low. Next, we'll look at the case of multiple alleles. So blood typing in humans, there are more than two allelic forms. We call them A, B, and O. And your blood type is determined by which two alleles are present, which leads to 4 different blood types. You can have type A, type B, type AB, or O. So an individual with type A blood has carbohydrate A on the surface of red blood cells. Blood type B has carbohydrate B. If you have blood type AB, you have both carbohydrates A and B. And if you have neither of those, your blood type is O. And you can see that in the figure 14.11b. You can see those are the surface carbohydrates that are sticking out. And if you look at A, when we do Punnett squares with these later, we don't use A, B, and O for the letters. We use the allele form that's on the top. But I'll remind you of that when we get there. We can also look at pleiotropy. Most genes have multiple phenotypic effects, which is what that pleiotropy is. Examples would be that pleiotropic alleles are responsible for the multiple symptoms associated with cystic fibrosis and sickle cell disease. So sometimes one gene has many phenotype effects. We also need to look at epistasis, where the phenotypic expression of a gene at one locus alters that of a gene at a second locus. Example here would be lab coat color. There is one gene that controls the coat color, so that would give us that part. And then there's a second gene that controls pigment deposition. So we get yellow labs because it doesn't really matter what the first gene says. If it says no pigment deposition, then we're going to have a yellow lab. And the last one is polygenic inheritance, where we have the effect of two or more genes on a phenotype. And this applies to lots of traits like human skin pigmentation. There are tons and tons and tons of varieties of that. And that's because of polygenic inheritance. Sometimes the environment has an impact on the phenotype. So it's going to be a combination of environment plus the genotype that it has. For example, in hydrangeas, depending on the acidity of the soil, it affects what color is shown. So if the soil is basic, the flowers will be pink. If it is acidic, they will be like a bluish purple. And the last part of this is pedigree analysis. Pedigree is a family tree diagram that geneticists can analyze to determine what traits might be present and whether they're dominant, recessive, or something else. We'll do some of this in class together because I think me talking through it might confuse you. We now have lots of genetic testing available to us, so we can find carriers of disease. So if a couple is thinking about having a child, but they have a history of a certain genetic disorder, they can get tested to see what the odds are of them passing that on to any offspring they may have. We can also do fetal testing. You can do an amniocentesis, where you take amniotic fluid that's analyzed, or a CVS, chorionic villus sampling, where a small sample of the placenta is taken to analyze. So that can tell you about the fetus before it's born. And there's also lots of newborn screening that can be done. PKU, that has to do with one of the amino acids, is a pretty common one. And there are picture examples of both amniocentesis and CVS.